All right, welcome back. Lecture number 6, we're going to go back to the testes, and now we're going to be looking inside the testes at a detailed level, and we're going to cover the process of spermatogenesis, which of course means sperm cell formation. We've got a multi-step process that has to go on here, and this continues in a male throughout his adult life. Okay, so again, this process of spermatogenesis refers to the multiple steps that are necessary to form sperm cells, and another term for mature sperm is spermatozoa. Spermatozoans would be, or spermatozoans, you'll sometimes see that as well. And this does take place, so if we cut open a seminiferous tubule in cross-section, remember you've got a lumen there, and that's where your fully formed sperm cells, they're not yet good swimmers, but that's where they end up in the lumen. And then over here in the wall of the seminiferous tubule, that's jam-packed full of cells that are undergoing this spermatogenesis process. And what we're going to see is the less mature forms of the sperm cells are out here near the edges, and then your mature sperm cells wind up there in the center. All right, before we get into spermatogenesis, and the concepts I'm getting ready to talk about are going to apply for egg cell formation as well, we need to review some basic information about the chromosomes of the body. Okay, so almost all of the cells, body cells, or somatic, that term refers to the body, contain 46 chromosomes. All right, chromosomes are DNA molecules. They have proteins that they're wrapped around to compact them down so that they'll squeeze into the nuclei of our cells. And these DNA molecules are the locations of genes. That's where your genes are located. All right, your 46 chromosomes are in a diploid state. All right, so diploid means you have 2 copies of each. All right, so you have 46 in the vast majority of the cells in your body, and it's 2 copies of 23 chromosomes. Okay, and you get one of those from your mom, and you inherit the other one from your dad. The 2 pairs are called homologous chromosomes. Homo meaning the same. Now, they're not identical because you can have, of course, differences in the genes that are located on these chromosomes when you're comparing the version you got from mom and the version you got from dad, but nevertheless they're considered to be homologous chromosomes. They're also numbered, so they're actually called chromosome number 1, number 2, number 3, number 4, up through 22. And then you have your 2 sex chromosomes, and you inherit one of these from mom, and you inherit one of these from dad as well. Those are your X and your Y chromosomes. And if you inherit 2 X chromosomes, one from mom and one from dad, you're going to be a female. If you inherit an X chromosome from mom and a Y chromosome from dad, you're going to be a male. And then that makes up that last pair of our 46 chromosomes. All right, so that's what's going on in the vast majority of cells in your body. Gametes, and again, that's a mature sperm or an ovum in a female. They only have 23 chromosomes, and that number is considered to be haploid, which just means you only have 1 copy of each chromosome. Okay, so a functional sperm cell, a fully formed ovum, only has 1 chromosome number 1, 1 chromosome number 2, 1 chromosome number 3, and so forth and so on. You still got to have 1 of each of those 23 chromosomes in order to have a full set of these, because those are going to have to be combined. If we're talking about the chromosomes in a sperm cell, those are going to have to be combined with the chromosomes from an ovum at fertilization to begin the formation of a new individual. So you got to have, it can't just be 23 random chromosomes that combine together inside a sperm cell. You got to have number 1, number 2, number 3, out to number 22, and then you've got to either have an X chromosome or a Y chromosome if we're thinking about sperm cells. All right, so again, just to review, and I know, unless you've had an introductory biology class or maybe you've already taken microbiology, you probably haven't had a lot of information on genetics yet. You will later. But as I was saying, the somatic cells of the body have 46 chromosomes. There are exceptions like your red blood cells. Your red blood cells, as we talked about earlier in the semester, they don't have a nucleus. So therefore, they don't have these DNA chromosomes on the inside of the cell. Your platelets don't have these either. And then you do have weird cells, like your skeletal muscle fibers that are multinucleated. So each skeletal muscle fiber actually has more than one nucleus. It's going to have more than one of these sets of 46 chromosomes. And again, a chromosome is a very large DNA molecule complexed with proteins. And this diagram is showing this to you. So here's a nucleus in a cell here, and we're pulling out a chromosome. Now typically when you see pictures or diagrams of chromosomes, they're drawn like that. It kind of looks like an X. And what that actually is a duplicated copy, two copies of the same chromosome, and it takes on this X configuration right before a cell is going to divide, and those two copies are going to be split apart. So just keep that in mind when you see that type of diagram. You're really looking at a duplicated pair of chromosomes, and they really only look like that before a cell is going to divide. And then if you zoomed really tightly into one of these chromosomes, you would find the typical double helix shape of a DNA molecule, that stretch of A's and G's and C's and T's that are linked together in two strands to form a DNA. All right, and then a gene, what do we mean by a gene? A DNA molecule is this long stretch of A, G, and C and T nucleotide building blocks. DNA is built from nucleotides. And along that DNA molecule, like let's say right there, and then maybe over here, you have a gene. Here's another gene over here, gene number 1, gene number 2. So they are actually just specific addresses or locations on a DNA molecule. And if you haven't learned it before, you will learn it in a later class. These genes on DNA basically function as blueprints that tell cells how to build proteins. Or you can think about them as recipes for proteins. And again, in another class, you'll learn more about how all of that works, but it doesn't hurt to become a little bit familiarized with it here. Now you have learned in Biology 201 that your proteins are the workhorses of your cells. Proteins, there are many different types of proteins. You've learned about many proteins throughout your 201 and 202 classes. So proteins, many of them are enzymes that perform critical chemical reactions. Many of them sit in the membrane of a cell and transport things across. Many of them are receptors for hormones. They have lots and lots and lots of different roles. Actin and myosin involved in muscle contraction, those are proteins as well. So if you can't make your proteins, or if you don't have the blueprints or the recipes for making all those proteins, a cell is not going to be able to function. That's why it's so critical that a cell have a full set of chromosomes. That way you have all of the genetic recipes that you need to build all of the proteins that you need to be able to survive and function. All right, let's briefly compare a couple of processes, mitosis and meiosis. So mitosis is the process that's used for typical cell division inside the human body. Meiosis is a two-staged special process for gamete production. So this occurs during spermatogenesis, and then when we cover the female, we're going to see that it's used in oogenesis as well. Let me erase all that. All right, so on the left-hand side over here, we have mitosis taking place. Now we're looking inside the nucleus of a cell, And if this is a human cell, you are going to have 46 chromosomes in there. But we're only going to take a look at sets of 2 chromosomes. So we'll say in pink, we'll say that's chromosome number 1, and in blue, chromosome number 2, for example. And then notice that they are duplicated. You have duplicated chromosomes. Okay, so we know that because they have this X-type configuration. They're kind of held together in the center, and that gives them that X appearance. And then these are colored on this diagram, blue and kind of pink. So that's supposed to represent inside that cell. The blue chromosomes were the ones, the versions that were inherited from this person's father. The pink ones were the ones that were inherited from this person's mother. So the DNA molecules, these chromosomes, these are the molecules of inheritance, as you guys know, since they have the genes on them. And those are the molecules that have to be passed down from generation to generation. All right, so during mitosis, now these chromosomes have been duplicated, and during mitosis, one of each duplicated chromosome is going to get pulled over into the 2 daughter cells that are made. So if you look closely at what's happened, let me erase all of that. For example, all right, so the 2 little chromosomes there, let's call those chromosome number 1. So those are each in a duplicated state. That means that DNA synthesis, DNA replication has already taken place. Those molecules have been copied, getting ready for cell division to occur. Same thing with these. Those DNA molecules have been copied and getting ready for cell division. When cell division actually takes place, those 2 copies that are held together like this get pulled apart. So one goes to one daughter cell and one goes to the other. Okay? And then that way you end up with 2 daughter cells right there and right there. And each one has 1 copy of each chromosome. So you're just seeing this for 4 of them. But remember that in the human, each typical somatic cell of the human body would wind up with 46 chromosomes at the end. Okay, some designations you guys will see in your textbook. This cell will be called 2N, which means diploid. And so will these. That's a 2N cell, and that's a 2N cell. Now you might be looking at that and thinking that doesn't look right, because this cell down here has 4 chromosomes, that one has 4, and this one really has 8. But because those chromosomes are duplicated copies of each other, you've got those replicated pairs that are held together in the center. A lot of times they're still considered to be 1 chromosome since they're identical copies of the same, and they're only going to temporarily exist like that until they get pulled apart. So that cell is still referred to as a 2N diploid cell. But in reality, keep in mind, you have duplicated copies of each of those chromosomes. So if this is a human cell, you have 46 duplicated chromosomes, and when you get down here, after the cell divides, you have 46 single copies of those chromosomes. Okay, now meiosis is very different. Remember, in meiosis, we need to generate, we're going to generate 4 haploid cells. All right, so instead of having 2 copies of each chromosome, we're just going to have 1. And this happens in 2 phases. All right, so in the first phase, which is called meiosis 1, the pairs of duplicated chromosomes, like your 2 duplicated chromosomes number 1 and your two duplicated chromosomes, number 2, they pair up with each other. And then in meiosis 1, only one pair of each one duplicated pair of each chromosome ends up in the first set of daughter cells. So these are your 2 daughter cells that you end up with after meiosis 1. Okay, and you have one set of duplicated chromosomes after the end of meiosis 1, if you look at that carefully. So that's different from what we saw over here. Over here, you had, for mitosis, you had 46 duplicated chromosomes. After the cells divided, each daughter cell had one set of 46 chromosomes. In meiosis, we're going to do 2 different steps because we need to cut that chromosome number in half. And again, this is called meiosis 1. And you're starting here, you've got 46 duplicated chromosomes. So that's a 2N cell, but the 2N chromosomes are in a duplicated state. And after meiosis 1, you go down to an N state, but the chromosomes are duplicated. They're still held together in the center, and they have that X configuration. Okay? Then for meiosis 2, meiosis 2 looks like mitosis. Okay, that's where the 2 copies of your duplicated chromosomes get pulled apart into each daughter cell. So at the end, you wind up with 4 cells that are N, or haploid. Okay, they just have one copy, if this is a human cell, of 23 chromosomes. You have one copy of chromosome 1 through 22, and then you have either an X chromosome or a Y chromosome. Okay, so this is a 2-stage process, meiosis 1 and meiosis 2, that's needed to get to the point where you have cells that contain, rather than 46 chromosomes, they just have 23. Okay, your textbook is going to have a lot more details on these processes, which you are certainly, if you want to read more about how all of this works, that's fine. I'm not requiring you to learn all the different stages of meiosis, like prophase 1 and metaphase 1 and anaphase 1 and telophase 1. You'll have to do that if you ever take an intro biology class, but we're not going to go that far with the information here, since we've already got plenty of stuff to learn with regard to the reproductive system anyway. All right, so spermatogenesis is going to involve 2 different things have to happen. One is we're going to have to do meiosis. We're going to have to do meiosis 1 and meiosis 2 so that we get to these haploid cells. And then there's a second part called spermiogenesis. So unfortunately, the names are very similar. Spermiogenesis refers to the processes of converting these cells that have a roundish spherical shape to something we would recognize as a sperm cell. So spermiogenesis is part of spermatogenesis. But when you see that term spermiogenesis, it's really referring more to a change in shape and structures. The meiosis part is over. All right, so we're going to see there are multiple steps in spermatogenesis. So there are cells that are called spermatogenic cells, and they are out at the edges of the walls of the seminiferous tubules. And they undergo regular mitosis. You have cells there called spermatogonia that are functioning like stem cells. So they haven't specialized in the sperm yet, and they haven't really started down that pathway yet, but they provide a source throughout a man's lifetime of cells that can undergo spermatogenesis. So those are called spermatogonia, and they periodically do mitosis, regular old mitosis, so they're still diploid cells that have 46 chromosomes, and you wind up with 2 daughter cells. One's called type A, and the type A ones continue to be spermatogonia. The type B ones are the ones that are going to head down the process of spermatogenesis. And they're also called, or they undergo some changes and become primary spermatocytes. All right, and then a primary spermatocyte is going to undergo meiosis 1. All right, so it's duplicated 46 chromosomes are going to split up into 2 daughter cells and be duplicated 23 chromosomes in each. And those 2 secondary spermatocytes, those are called secondary spermatocytes, they undergo meiosis 2, and that's going to generate 4 cells that are called spermatids. All right, so the secondary spermatocytes have 23 duplicated chromosomes, and when those cells divide, you end up with 4 cells, each of which just has 1 copy of each chromosome. Those are haploid cells. And so now we have the correct number of chromosomes that we need for reproduction, for fertilization of an egg. The spermatids, though, have a roundish shape, and so then we need to do this spermiogenesis process. So we'll have change in shape and structure so that those cells can have everything they need to do the swimming that's required for fertilization. Okay, so spermatogenesis is a long process, takes 64 to 72 days. It continues throughout a male's lifetime, generating gazillions and gazillions and gazillions of sperm. But it does not begin until a male reaches puberty, because as we'll see, this process requires testosterone production, the male sex hormone, for stimulation to make it happen. Okay, and here's a diagram which is showing you, this is a wall, oops. You're looking at the wall of the seminiferous tubule here. And then down in here is the lumen. All right, so this is the edge of the wall, and down in here you would have your lumen, and so right in there, that's supposed to represent one of those spermatogonia I was talking about. That's a stem cell, and that's a 2N. It's a diploid cell, and it undergoes regularomitosis. And when that happens, you wind up with a type A daughter cell and a type B daughter cell. As you see there, the type A daughter cell just continues on as another spermatogonium. It's going to be a source of continuing to supply these cells that are needed for spermatogenesis. The type B daughter cell, now it doesn't divide, it just changes and undergoes some specialization to become a primary spermatocyte. All right, so that's still 2N. It undergoes meiosis 1 and generates 2 secondary spermatocytes. And now you're actually considered to be a haploid cell, but just keep in mind that those 23 chromosomes are still in a duplicated state like this. Each of those cells divides and gives you 1, 2, 3, 4 early spermatids. And those are haploid. And when the secondary spermatocytes divide, their duplicated chromosomes are pulled apart and into opposite daughter cells. Okay? So those are your meiosis stages and the cell types where they take place. The rest of what happens to form sperm is called spermiogenesis, and it involves taking these round cells you see here and converting them over to these cells that we recognize as sperm. Okay, and I believe, let's see, that slide just has text written out for you, which is what I just went over. And this is a diagram from your textbook, figure 27-8b, which also, again, is reviewing, just without all the background parts of the diagram from the textbook there, showing you what I just talked about. Here are the cells that are actually involved in meiosis. And then here's your spermiogenesis down here, where those early spermatids that look round become late spermatids, are starting to take on the physical characteristics of sperm. And then finally, you have your spermatozoa. That's another term for a sperm cell. The spermatozoa have all their sperm cell parts, but they're not fully functional. They're not good swimmers. They have to pass through the epididymis before they fully mature into good swimming sperm. Let's see. I see a typo on this slide. That should be spermatozoan rather than zoon with two O's. Okay. Spermiogenesis involves multiple steps. Spermatids have to elongate because they're starting off as round or spherical cells. They have a lot of excess cytoplasm because they have to develop that sperm head. And the sperm head has the 23 chromosomes inside it. They also have to develop the tail or flagellum. And once all that has happened, then you have your sperm cell. Here's a nice diagram from your textbook which gives you a better impression of these changes in structure that occur during spermiogenesis. So again, this is the spermiogenesis process. And now you don't have to memorize all these different changes, but you can see how the sperm head is much smaller than the spermatid that emerges after meiosis too. And that sperm head on the inside, you can see the nucleus. That's where your 23 chromosomes are going to be. Then you've got this structure, the acrosome, that kind of looks like a helmet that's sitting on top of the nucleus of the sperm. One of the things that the acrosome contains is enzymes that are going to help the sperm penetrate the egg cell, which we'll be talking about later during the unit when we cover fertilization. This sperm cell also has a midpiece in here, and there are lots of mitochondria in there. Mitochondria are the sites of the aerobic oxygen gas requiring steps of cellular respiration, so they're very much involved in generating ATP, and sperm cells are going to need lots of ATP for swimming. And the tail is also called the flagellum. And I think I just covered all of the info that's summarized on the text here. Again, the midpiece section has lots of mitochondria. Those are organelles where most of cellular respiration takes place to generate ATP. Okay, another cell type in the walls of the seminiferous tubules, and let me back up because I forgot to mention these when we were back over here on this slide. Okay, so we were talking about all these cells that are involved in spermatogenesis, but if you look, all the yellow stuff you see in here, all over the place, more of it over here, that is cytoplasm from very large cells that are called cystinocytes, or Sertoli cells. That's an older term for these, or cystinacular cells. Those are all names for those. So these are very large cells that are make up, you know, a fair amount, if not the bulk of the wall of these seminiferous tubules. And they're big cells, so this is one cell all the way from one end to the other. And they wrap around and surround these other cells that are involved in spermatogenesis. And on your diagram too, they're showing you like right in there, and I don't see them anywhere else on the diagram, but that's indicating there are very tight junctions between these cystinocytes or Sertoli cells in the wall. So it helps hold the wall together, and parts of these Sertoli cells just wrap around these other cells. Those other cells are embedded within those. And let's see, let me go back to where I was. Oops, okay. All right, and what are these Sertoli cells? I mentioned some of their roles. They do extend all the way across the wall of a seminiferous tubule and surround the other cells. They have lots of roles. They provide nutrients. They provide signals to the dividing cells, so they're involved in some of the communication, telling these cells to undergo meiosis and make changes. They're thought to help move the cells in the correct direction, where you have the more immature spermatogonia out near the edge of the wall, moving the more mature ones toward the center, toward the lumen. They secrete testicular fluid, the fluid in the lumen that the newly produced sperm cells are floating around in, that's secreted by these cystinocytes. They're also phagocytic, so when you have germ cells here, refers to, that term is referring to these developing sperm cells. That's another phrase that can refer to those, like the germ line, things like that. So if you have any of these cells that are faulty, errors have been made, and they're not going to be functional, these cystinocytes can phagocytose those. Also, they're the ones, the developing sperm cells have to lose quite a bit of their cytoplasm to take on their correct shape and structure, so the cystinocytes are going to gobble that up as well. And they do produce chemicals that help regulate these processes. Another thing they do, those tight junctions we were taking a look at, and tight junctions, again, that refers to their proteins that form almost like snaps between cells and help hold them together. And so that wall is all very, very tightly compacted, and you don't have blood vessels in there. It is a type of epithelial tissue, so your cells are jam-packed tightly together and you don't have blood vessels passing through the wall. And the cystinocytes help maintain what's called the blood-testis barrier. And interestingly, that's important because if we didn't have that blood-testis barrier within the walls of the seminiferous tubules, if you think about it, sperm cells don't develop in a male until a male reaches puberty. So the immune system has never seen some of these sperm antigens, some of these sperm molecules have never been detected before by the immune system. So if sperm gets into the blood, then the immune system would learn to recognize these sperm as foreign to the body. The immune system is going to think this is some sort of an invader, like a microbe, and then you will develop immune responses to sperm. And those sperm would wind up getting attacked and killed by the immune system, and that could lead to infertility. All right, and speaking of infertility, we've all heard of males who are infertile. Now there is a gradual decline in male fertility past the age of 50, but we all know that there are men who father children even beyond the age of 80. So sperm cell production continues in a male throughout his reproductive lifetime. But sometimes sperm counts drop below where they're supposed to be. And what's causing this? There are many different causes. Xenobiotics, that's a term for alien molecules. Xeno refers to something that's foreign. So it's thought that, in our society today, we're exposed to a lot of toxins, PVCs, chemicals that are located in plastics, pesticides, herbicides. There are chemical compounds that we get exposed to that are chemically very similar to estrogen. Some of those are present in plants as well. Some of these types of chemical compounds are thought to leak out of plastics that get microwaved. There's been some information in the news the past few years about, if you're a male and you don't want to be infertile, you might want to not use plastic microwave cookware because some of these xenobiotics that may maybe leaching out of the plastic microwave cookware and playing a role in infertility. A lack of selenium. Selenium is one of those mineral nutrients that we need in very small quantities in our diet because it's likely a cofactor for an enzyme or enzymes needed for sperm production. Excessive alcohol intake can be toxic for sperm. There are some men who lack a specific calcium channel, so that would be a genetic type disorder. There can be anatomical obstructions, hormone imbalances, high fevers, hot tubs, because the temperature gets too high in the scrotum for the testes if you spend a lot of time in hot tubs. So lots of things can lead to infertility, and a lot of times it's just a matter of trying to figure out what the root cause is and trying to correct that. Okay, so that was a fairly long lecture. Lecture number 7 might be fairly long as well because we're going to talk about the hormonal regulation of male reproduction. What stimulates spermatogenesis and how is it kept under control, negative feedback and so forth. That'll be the subject for lecture number 7.